All right, welcome back to the Woodshed, aka Hanson Studios. So we're going to start the next lesson here, and this has got to be one of the biggest ones. Let me go ahead and switch my camera over. We're going to be looking at proteins now. That's what this chapter's titled. So 16.4 is about proteins. So we got a lot to talk about. We've done a lot of the groundwork talking about amino acids and how they come together to make peptides. But proteins are essentially polypeptides or many peptides together, right? But it's not just a bunch of polypeptides coming together. Proteins really take shape. They really take shape, and they do a lot of functions and a variety of different things based on their shape. And so that's what makes proteins pretty cool and unique, is what shape they take. And so that's kind of what this lesson's all about. It's about proteins and how they take their structure. Notice you see structure. So we're going to analyze what the primary structure of a protein is, what makes the secondary structure of a protein, and then finally what contributes to the tertiary and quaternary structures. So tertiary means 3, by the way, and quaternary means 4. So here you go. Let's jump right in. I'm going to connect to my PowerPoint here, or Keynote. So I got the Keynote pulled up here on my laptop. Let me get that going here. It's going to be my camera 4 input. So there's the tunnel of doom until I minimize that screen. And there's me at my wedding. Nice tux there. And let me just pull up the Keynote. Here you go. All right. And so one thing I also want to do is show you this slide that is pretty cool. I found this online. It talks about the coronavirus and how it has these spike proteins, and those proteins do what they do. And anyways, I'll go into that when we get towards the end of the lecture, just for your pleasure. But we're up here at 16.A, primary structure. So 16.6 is again about proteins. And so to understand proteins, which are those large polymers of amino acids, and amino acids would be the monomers of the polymer, you have to learn about their 4 levels of structure. That's what makes proteins so special, is these 4 levels of structure. And so starting with the primary structure, and real quick, it says they're responsible for so much of the structure and function of all living organisms. I can't stress that enough. I mean, if you've taken my biology course, then you know what I'm talking about. You know the DNA can only do one thing, and that is that it is the code. It contains the code for which the ribosome can use to make proteins. And so, that's a pretty big deal. That's a pretty big deal for you to be who you are. You started out as one cell with just a bunch of DNA, and that cell had nothing unique. It didn't have any earlobes or eyeballs or anything like that. It was just a cell. And so for your cells to do what they do, they've differentiated, they've copied themselves, and they've become different cells, and then they communicate with each other, and it's a phenomenal process. And for all that to happen, the DNA has it stored. Your DNA has everything about you stored in it. And so that is an interesting and amazing feat. And so let the dog in here. So he's going to join us for this class. Okay, so back to the lesson here. We have the primary structure. It's got to be the most basic. It's pretty much what we've already learned. And that is that it's the particular sequence. Highlight that word, sequence. The primary structure of a protein is basically that sequence of amino acids that's joined together. Okay? Whether it be 16 to 20 to 30 to 40 to 50 amino acids, however long that chain is, whatever that sequence is going to be is super important. And remember, that's what the DNA does, is it contains codons that the ribosome reads one at a time, and each codon corresponds to a specific amino acid. And so, first of all, that primary structure is going to be very specific. And even if even one of those amino acids is out of place, you're going to have a different protein eventually. But starting out, you just have a sequence of amino acids. Like it's not even a protein yet. You're going to see there's going to be a lot more shape, a lot more taking shape to the protein after this. But again, that shape is dictated by this specific sequence. Now, something to point out. If you were to just simply take all of the amino acids and put them in a row next to each other without doing any of the further discussion we're about to do, you would simply get this pattern here. You would get an amino acid right next to an amino acid, right next to an amino acid, and from far away it would just look like a straight line. It would just look like a straight line because check it out. The geometry of these bonds here, if you look at this carbon bond, if you look at this peptide bond or this amide bond, this carbon-nitrogen bond, every bond here to here to here to here to here is all trigonal planar. If you forgot what that means, it means that all of these bonds lie in a plane. So you could literally, you're looking at the three-dimensional structure here. Even though it's on a two-dimensional page, this is actually the three-dimensional structure. This is what this would look like in real life. If you took the ball and stick model and laid it down on the counter, these would all be in the same plane. Nothing would be poking up above the table. Nothing would be hindering it from laying flat on the table. Like this would be, and all these angles are 120 degrees because that adds up to 360. And so you have trigonal planar geometry there. Okay, so all 6 of the atoms involved in this peptide bond, 1, 2, 3, 4, 5, 6 atoms, all those atoms are participating in bonds and they're all lying in the same plane, all right? And so that, as a result, that ends up meaning that the backbone of a protein adopts this zigzag arrangement here. And from far away, it just looks like a long straight line of amino acid after amino acid, all right? And then the R groups of the amino acids will poke up and down like this. They'll be aiming down and then up and then down and then up. And those R groups, remember, are the variable groups of amino acids. And so these R groups are really going to they're really going to make an influence on the shape of the protein later on, which we'll talk about. But for now, if you were to stop here, what we've discussed so far, you would have the primary structure of a protein. The primary structure is just simply the particular sequence of amino acids, okay? And again, you pretty much just have a straight line until, I mean, it's not going to stay this way, but I'm just saying if you were to stop here, you would just have a straight line of amino acids, and you'd have all these, you'd have this zigzag like this. Again, I'm just saying, like from far away, you would just see kind of a straight line. And the primary structure of a protein is the exact sequence, okay? It's going to eventually determine all the properties and functions of a protein, because you'll see why. If you just substitute one single amino acid for another one, you'll end up in a different protein. So let's move on to secondary structure. The secondary structure is where you finally start to get some shape taking place. And basically, it's the three-dimensional arrangement of localized regions of a protein is going to be your secondary structure. It's going to be the three-dimensional arrangement of localized regions. And so these regions arise due to what's called hydrogen bonding between the NH proton of one amide and the CO oxygen of another. So the hydrogen and oxygen basically are going to be attracted to each other. I'm not going to say bonding. I'm going to say they're attracted to each other. We call it hydrogen bonding, but it's not really bonding. It's not like covalent bonding where the electrons are shared. You simply just have kind of an attraction. This happens with water. Those magnets I always use in class. Those magnetic water molecules. Because the hydrogens are essentially just a proton, because oxygen is sharing the electrons, holding on to the electrons longer because it's an electron hog. Then you essentially on a water molecule kind of just have two hydrogens by themselves as just protons. They get an electron every now and then. They get to share it, so they're sharing. It's a covalent bond, but that part of the water molecule is essentially just a positively charged proton, and the oxygen's there like the big hog, hanging on to the electrons for a long time, and he's essentially an electron hog, and so he's going to have a negative charge because electrons carry a negative charge. And so water molecules have a region of positive charge and a region of negative charge, making it polar, just like a magnet would be. And so that magnetic molecule kit I have is used to show that. I even brought it from home, but I'm not going to worry about pulling that out now. So that's kind of what's going on here, but it's not with water molecules. It's with these proteins. Let me just double check my... Okay, I just wanted to see things were going good. All right. So we just have hydrogen bonding taking place. Now, when that hydrogen bonding takes place between the NH and carbonyl of another, then you're going to get these type of arrangements. You're going to get, and don't worry, it's just two. You're going to have an alpha helix or a beta sheet, a beta pleated sheet to be exact. And so we're going to go into discussion now about what an alpha helix is and what a beta pleated sheet is. All right, this is huge. This is going to be taking place all throughout the polypeptide chain, and this what really dictates a lot of the structure of a protein. And so the alpha helix forms when a peptide chain twists. When it twists into a right-handed or clockwise spiral, as shown in Figure 16.4. I know it's kind of hard to see here. That's why they've had to. This is on two-dimensional paper, and they're trying to show you a three-dimensional like twisting here, almost like you're going up a spiral staircase. You know, you're going around and then around and then around and then around. So it's a spiral. It's a helix. It's a twisting happening, and the way this is taking place is that the carbonyl, or the carbon double bonded to an oxygen group of one amino acid, is hydrogen bonded to an NH group 4 amino acid residues further along the chain. So if you look at this picture here, you'll see the carbonyl groups pointing up like this, and you'll see the NHs pointing down. And when you go 1, 2, 3, 4 amino acids down the chain, you'll see there's an attraction there. If we go all the way down to the bottom, we can see it again. You see the carbon oxygen pointing up here, and you'll see this nitrogen and hydrogen pointing, or the nitrogen and hydrogen pointing down. This nitrogen and hydrogen is attracted to this carbon and oxygen. This nitrogen and hydrogen is attracted to this carbon and oxygen. I know it's kind of hard to see because they've thrown a lot in here, but the only thing I need you to really get and understand is that it is that carbon double bonded oxygen that's attracted to the nitrogen bonded to the hydrogen of another amino acid. And it's not a bond that necessarily takes place, but it's just an attraction. All right. And then the R groups, the variable groups of all the amino acids, they extend outwards from the core of the helix. All right. So all these R groups, they're poking out to the left or they're poking out to the right of the helix. Do you see that? None of the R groups are pointing into the helix. They're coming out from the core. You see this R group here that kind of looks like it's in the helix. It's really coming straight out at you. All right, and there could be another one over here, running away from the helix. But the point is, the R groups extend outward from the core of the helix. All right, so there's your alpha helix. Remember, it's when one amino acid is attracted to another amino acid four bonds away. So you count 4 amino acids, and there you go. The first amino acid will be bonded to the fourth, and then 5, 6, 7, 8, 9, 10, 11, 12, right? Every 4 amino acids, you'll get an attraction to each other. Every 4 amino acids, you'll find an amino acid that's attracted to the next amino acid 4 amino acids away from it. All right? And again, it's the nitrogen and hydrogen that is attracted to the carbon and then the oxygen. And maybe in another lesson I'll try to find a better picture of that, because this one is kind of weak on that. But point is, speaking of weak, those are weak bonds. They're weak bonds. It's hydrogen bonding, so it's not necessarily super strong. But that hydrogen bonding in that particular format will give you an alpha helix. Now, the beta pleated sheet is also due to hydrogen bonds, but in a slightly different way. The beta pleated sheet forms when 2 or more peptide chains altogether, called strands, we call those strands, when 2 or more different peptide chains are attracted to each other, or they line up side by side like this. Like you have a peptide chain here, peptide chain here, a peptide chain here, a peptide chain here, and they're all lining up like side by side like this. Then they form this pleated sheet. They form this pleated sheet, and it's also, again, due to hydrogen bonding that is occurring between the, you guessed it, nitrogen and hydrogen of one amino acid and the carbon double bonded to an oxygen of another amino acid residue. All right, now, and then the R groups in this case are pointed or oriented above and below the plane of the sheet. All right, so if you were to look at this sheet, you'll see the R groups in yellow. This is not sulfur, by the way. They're just putting yellow as the color, which they shouldn't have done, but hey, because yellow is usually sulfur. But these R groups. They're pointing up above the sheet, right? There's a group of R groups pointing up above the plane of the sheet, or they're down here aiming down below the plane of the sheet, like they're pointing down. All right, so there you go. You have your alpha helix, or you have your beta pleated sheet. Both are due to hydrogen bonds. And in both cases, you essentially end up with R groups either like coming away from the core or coming up and down below the pleated sheet. All right. So there's ways that we show that. Proteins are huge. Proteins are huge molecules, and so if you try to do a ball and stick model like this, it's kind of ridiculous. Like here's a lysosome or lysozyme. Lysozyme is an enzyme that helps break down the cell walls of bacteria, and so it's needed to do that. But look how big this is. I mean, yes, it can help you see, ooh, I have an amino acid here, an amino acid here, and an amino acid. You could look closely and figure that out, but there's a lot of amino acids going on there. And so is that all necessary? No. What's necessary is what are the R groups on the amino acids and how is it all taking shape? So rather than doing a ball and stick model or rather than doing a space filling model, it's so much easier to do these ribbon diagrams. So people that are into molecular biology and do that sort of work, if you look in any textbooks, you're going to see stuff that looks like this. In fact, here, let's just do this. Let's do a protein structure Google search and check it out. You're going to get these crazy ribbon structures like this, or like this, right? Look at that, crazy structures. You'll get stuff like this. I mean, it's almost like artistic looking. You see like the ribbons, right? You see the alpha helixes and you see the pleated sheets, right? Really exotic stuff. I mean, it's extremely artistic. Look how these are huge molecules, right? And what you can see is you can see ribbons and coils. There you go. There's a virus coat protein. Oh, look, you got some animation there. Oh, isn't that neat? It's like a one of those like pinwheels, that have like the little blades. Isn't that awesome? I wonder how many of these like 3-dimensionals I could get like that in animation. Oh, nice. That looks like a mix between a space filling model and a ribbon diagram. But yeah, again, proteins are huge, and so there's just a vast, almost infinite amount of proteins that exist, and so you're typically going to see these ribbon diagrams, so you can keep it easy. All right, and you can kind of look at specific regions and see what's going on. And so again, this right here, this little ribbon, the ribbon that has the let's see, flat helical ribbon. I'm reading this right here. A flat helical ribbon is used for the alpha helix, while a flat wide arrow like this, a flat wide arrow, is used for the beta pleated sheet. And then this is just used to represent random, not really alpha helix or not really beta pleated sheet. It's just These are just amino acids connected to each other, but not really in any kind of special particular way. And so proteins are drawn in a variety of ways to show different aspects of their structure. And figure 16.5, which we'll look at in a different, and it shows, that's up here, we already looked at that, shows the three different representations of protein lysozyme, an enzyme found in both plants and animals. It catalyzes the hydrolysis of bonds in bacterial cell walls, weakening them, often causing the bacteria to burst. All right. So again, these are called ribbon diagrams. That's typically how you're going to see proteins. Let's read this part. This is important. The beta pleated sheet arrangement is favored by amino acids with small R groups. So the beta sheet like this is favorable for amino acids that have like small R groups. For the small side chains, if they're small, the beta pleated sheet's pretty favorable. However, if you have large R groups, then you're going to get steric interaction. You remember steric interaction from organic chemistry? It has to do with molecules that just take up a lot of space, and they don't really like to be around other molecules that take up space. So everything likes to be as far apart as away from each other as possible. That's actually why you get this arrangement here. If you have three atoms, what's the only arrangement they can be as far away from each other as possible? It's to form these 120-degree angles between each other, right? And so steric interactions not going to be very steric interaction is going to make this not very convenient for large side chains. If these yellow R groups here, the side chains of all these amino acids, if they're large, then this is going to be favorable. The pleated sheet is favorable because you got plenty of space above the plane or below the plane of the sheet. The helix, however, is not going to be very favorable, because how are you going to try and get an R group side chain all sandwiched inside that helix, right? It's not going to be very favorable. There's not going to be enough room for that. And so with larger R groups, steric interactions prevent the chains from getting close together, so the sheet cannot be stabilized by hydrogen bonding. All right, so the beta pleated sheet arrangement is favored by amino acids with small R groups. With larger R groups, steric interactions prevent the chains from getting close together, so the sheet cannot be stabilized by hydrogen bondings. So I just said it wrong again the second time. This sheet is favorable for small R groups, but for large R groups, you can't pack it all in like this. Like, there's actually not a bunch of room up top, like I said there was. So that was the little mistake I just made. Sorry about that. You don't have a whole lot of room up here, because this is the only place where they can all go, is up top. Well, if you got a guy up here that's really big, he's going to be taking up a lot of space. And so large side chains aren't going to be very, you're not going to see large side chains in amino acids that are participating in like a pleated sheet. It's just not going to happen. If you see a pleated sheet, it's pretty safe to assume that you got a bunch of small side chains in a lot of these amino acids. And so can you imagine whose job it is to sit and look at protein structure or primary sequences and figure out, okay, this bond is going to happen here, this bond is going to happen here, and come up with all these structures? That's somebody's job. There are people that literally walk through the process and say, okay, we got this polypeptide, so then it must make these, which are eventually going to make that. And it's like, that would be a lot of work. Now, it might eventually be fun once you get good at it, but it seems like at first it's going to be a lot of analyzing thousands of amino acids. Okay, so now the last levels, I should say, this is the last section, 16.6c, and it has to do with both of these structures here, or both of these levels of structure, tertiary and quaternary. Now they kind of go together because these are the primary and the secondary are the main contributors of structure. Primary structure is huge because it has to do with the sequence, right? It's a big deal. Secondary structure is huge as well because it's what's allowing all of the helixes and all of the sheets to come together. Once you know that, then the tertiary structure is just simply the final shape that takes place, or not the final, but next almost final shape that takes place. It's basically the three-dimensional shape that's adopted by the entire peptide chain. So once all of the sheets have happened, once all of the helixes have happened, then you're going to get this kind of overall shape, which is going to represent the tertiary structure. A peptide generally folds into a shape that maximizes its stability. So remember, everything wants to be as far away from everything as possible, unless there's, a couple atoms that want to share electrons, which that can happen, which we'll talk about in just a second. Then you'll have that happen to kind of bring things together a little bit. But once everything that's happened that should happen as far as interactions go, you're finally going to get your shape, which is going to be the tertiary structure. And then in the aqueous environment of the cell, proteins often fold in such a way as to place a large number of polar and charged groups on their outer surface to maximize the dipole and hydrogen bonding interactions with water. So a lot of proteins, if you were to look at the external surface of the protein, the kind of the perimeter of the structure, you're going to see a lot of polar regions so that hydrogen bonding can occur. Dipole is kind of the same thing as hydrogen bonding. It's negative and positive attractions, not bonding, but like attractions. And so in the body, when proteins are in water, in solution, then they're going to kind of arrange themselves in such a way that a lot of those polar groups, like hydroxyl groups or NH groups, they're going to be on the perimeter so that they can be dissolved in water pretty well. And then that generally places most of the non-polar side chains in the interior of the protein, where London dispersion forces between these hydrophobic groups help stabilize the molecule too. So London dispersion forces are like just the weakest forces you can get. And that's basically just like you have matter, it's attracted to matter, but not in any kind of charged way. If you have atoms or material, it's just going to be attracted to other material. Okay. Let me show you what London dispersion forces are. They're very weak, very weak. And basically every other attraction will overpower a London dispersion force. But what I wanted to find was the gecko example. The way geckos stick to surfaces with their feet is because they have these van der Waals forces, which are essentially London dispersion forces. And basically the feet of the gecko, they have so much surface area. Like if you look at it under a microscope, you'll see all these little tentacles, and that creates so much surface area that you end up with an attraction, and it's sort of an adhesive attraction, but it's not really from electrical charges like positive and negative. It's just the fact that you have all this surface that's going to be attracted to the next surface. They're able to climb any surface due to those London dispersion forces between the feet and the surface. Cool. All right, so going back to our slide here, if you have something like this, if you have your sequence, then you can start making predictions. Again, I know this would be really kind of like annoying to do with like super large molecules, but insulin's a fairly small protein. So let's look at this example, this figure. Insulin is just a small protein consisting of two polypeptide chains designated as the A and the B chains. So here's your A chain that has, let's see, what was it? 21 amino acid residues, and the B chain has 30 amino acid residues. Oh, the numbers were here. I didn't have to scroll down. Sorry. 21 and 30 amino acid residues. You have your A chain and you have your B chain. All right. And then they're held together by what are called disulfide bonds. All right, so this is a very special bond here between cysteine and cysteine. And I think we covered this in the last lecture. Cysteine has an SH group, which is sulfur and a hydrogen group, as its side chain. And that will actually covalently bond with another cysteine's sulfur group. All right, that's like a real bond. That's a real connection. Super strong covalent bond. And so what's happening here is the A chain is being bonded to the B chain through this cysteine and this cysteine. So you see that disulfide bond? Sometimes that's called a disulfide bridge. A disulfide bridge. There's actually another one right here. Cysteine is bonding on the cysteine on the B chain is bonding to this cysteine on the A chain via a disulfide bridge or disulfide bond. All right. And then just as a coincidence, there's also a disulfide bond even just on the A chain in and of itself. Like this cysteine on the A chain is bonded to this cysteine on the same A chain. All right, that's going to give it a lot of shape. Insulin, as you know, is used to control blood sugar. It's manufactured in the pancreas and in these islets of Langerhans in your pancreas, these special cells that make up the islets of Langerhans inside your pancreas, and they're going to make insulin. And insulin's going to be good at grabbing onto glucose and allowing it to get into the cell. Glucose can't just enter cells. Glucose is a fuel for cells, but it can't just enter directly. It needs a bodyguard, or it needs like somebody to carry it into the cell. The cell won't recognize carbohydrates like glucose and just allow them in. It needs insulin to do that. And so if you don't have insulin, guess what? You just got a bunch of blood sugar building up. Like you got a bunch of glucose in your blood, and that's bad. So you need to get that blood out. So you'll have to oftentimes have to inject insulin so that you don't end up with negative side effects. Okay. In addition, polar functional groups like the carbonyls or carboxylic acids, carboxylate ions, I should say, and these ammonium cations, they can also stabilize tertiary structure by electrostatic interactions. So when you have like a negative charged region of an amino acid and a positive charged region of another amino acid, they can be attracted to each other. Then those electrostatic interactions are kind of like a hydrogen bond, basically. Okay, they're kind of like a hydrogen bond, and that'll contribute to the overall shape. Finally, well, we just said this, but finally, disulfide bonds are the only covalent bonds actually that stabilize tertiary structure. So when you get amino acids attracted to other amino acids, that's not a real covalent bond, they're just an attraction. But when cysteine and cysteine form that disulfur bridge, that's an actual covalent bond again, okay? Just reinforcing that. Okay. I already went through the insulin example here with the two chains of insulin and how they're bonded to each other. So here's the final structure, or final contributor to the protein structure, which is the quaternary structure. And that is the shape that's adopted when two or more folded polypeptide chains come together. So that can actually happen. So you can get everything that we've discussed so far happen to some polypeptides. And then those polypeptides can come together with each other. So to form like kind of complex structures. And that would be a quaternary structure of a protein. And so in that case, each individual polypeptide chain would be like a subunit of the overall protein. A perfect example of that is hemoglobin. Hemoglobin consists of 2 alpha and 2 beta subunits held together by intramolecular forces in a compact three-dimensional shape. The unique function of hemoglobin is possible only when all 4 of those subunits are held together. So here's hemoglobin. In a quaternary structure, you have this subunit, this subunit, this subunit, and this subunit all held together. And then actually there's an iron atom in the center there. I'm not sure why they didn't show that. And that's what helps attract the oxygen to hemoglobin. But I love this picture. You just basically cut this out of your book with, no, you don't have a book, but cut this out with scissors and put it on your wall, or screenshot that, or circle it. This is a really cool picture that shows you all of the different interactions that could take place. So you have hydrogen bonds that could take place this way, hydrogen bonds that could take place this way. You have electrostatic attractions that are kind of like hydrogen bonds between charged regions of side chains, R groups. You have London dispersion forces, which is literally just non-polar regions attracted to each other. Not in an electrostatic way, like positive attracted to negative, but just non-polar stuff attracted to other non-polar stuff, just because the fact that they're material, they're atoms. And then you have hydrogen bonds again that can form like these pleated sheets. You have hydrogen bonds that can contribute to this alpha helix. Remember the helical structure? Then you have disulfide bonds that are actually like covalent bonds that can literally make this thing wrap all the way around and connect right there. That's strong. You're not going to undo that. And so these are the four levels, or the 4 levels of protein structure are basically summarized in 16.8 down here. So figure 16.8, this kind of summarizes it all. This is a nice picture to study. I'm not going to go over it with you right now. In fact, I think this video is long enough. I don't really want to go into all this. I'm going to make a video on the coronavirus and how it works, and how these spike proteins are recognized by the cell's receptors here, and then allow it to come in, and then the RNA is translated and turned into these proteins that end up being those spike proteins again, and hijacks the cell to make these proteins, hijacks the RNA, hijacks the ribosome and its RNA-dependent RNA polymerase to copy these and make more proteins again, and basically you're just going to keep making these viruses over and over and then sending them out, and it causes a immune response in the cell. But it looks like you have a couple problems here you could focus on, or that you could work on to test your skills. Let me know if you have questions on that. The next lesson will be about focus on the human body again, but so we'll actually talk about specific proteins like keratin and whatever. So all right, so that's going to be it for this lesson. A lot was there. If you need to, pause, rewind it, go back, rewatch it. There's also a lot of stuff on Google Images if you just type in protein structure, and you can kind of do a lot of your own independent research there and find some cool stuff about, or cool diagrams to study. And you might find a lot of other YouTube videos too that go into this that you might find a little bit better quality than mine. I don't know, but I did walk you through the book, which we're using, so hopefully my video is directly helpful for you guys. And as always, thank you for watching.